Okay, let's begin. Translation. I know you're tired.
No,
Let's
we're
start
not.
with an animation. Have you noticed I've been using a lot of animation, at least for this? So visit those links. I've been sharing the links with you. I think let's not watch this. This is pretty short. So I have your DNA inside the nucleus, and then transcription needs to happen first to produce your messenger RNA. Remember, this messenger RNA underwent 3 modifications, right? Splicing
Capping.
and tail. All right. Now it's your final mRNA. It needs to be translated into your protein. Okay? So the code is transcribed, which is translated. I've mentioned this before. This is called a transcript. Transcript is like a copy. And we call it translation because if it's mRNA, it doesn't sound like anything. It's like blah, 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 blah. The cell doesn't understand it. It needs to be translated first into something that can be understood, which is your protein. And protein can be understood because it has a specific function. So it will be translated into a molecule that will be easy to understand. Now, how does translation proceed? Okay, maybe now some movement.
Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Okay, so you have that moving picture in your head now. All right, let's go into the details. First of all, I was expecting you to ask this question. Where does this come from? Or this is your codon, which accords with I'll just point here so Enzo can see. This is your codon, this is your anticodon. And then last class I said this anticodon has a specific amino acid there. A different anticodon will have a different specific amino acid. So we'll have to look at the structure of your tRNA first. Now, this is the structure of your tRNA. I use this picture so that you'll think, remember, it's still an RNA. An RNA is single-stranded. Look at the structure. This is single-stranded. So you see the 5' right there. And then loop, 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 and then the 3' end. So it's still one long single-stranded, it's still one long single strand. It's just looped in 4 corners. That's why it looks like a letter T. But it's still just one strand, right there. One strand. And what's important for you to know here is that the specific amino acid is attached on the 3' end. Okay, we've been emphasizing 5 prime, 3 prime, 5 prime, 3 prime. Don't forget this. The amino acid is on the 3 prime end. Okay? Now, what you need to know is, how is this attached at the 3 prime end? What we call an enzyme called the tRNA activating enzyme. Only one Filipino here. I always joke about this. It's called tae. Tae in Filipino means poop. That's right. T-A-E. tRNA activating. That's important. tRNA activating enzyme is the enzyme that binds this amino acid to a specific tRNA. Look at this diagram here. So you have this enzyme. Let's say it's the amino acid is valent. It needs to be attached to a tRNA using, what is this molecule? ATP. So with the use of ATP, ATP gives off energy. So remember, removal of phosphate. I said removal of phosphate liberates energy. So when you break this here, it gives energy to the enzyme it gives, how do I, how do I remove that? So annoying. Oh, okay. So the energy from the hydrolysis of the ATP gives energy to the enzyme to bind that amino acid with an incoming tRNA. Okay? So ATP is used. Hydrolysis of ATP gives energy to bind this with this. So this is now your product. Are enzymes specific?
Yes.
Okay. So how many amino acids do we have?
20.
20. We have 20 different amino acids. So, do you expect to have 20 different taes?
No. What? 20.
Yeah. Enzymes are specific to their chemical reactions. You need specific amino acid with specific tRNAs. So you also have 20 specific tRNAs, right? So, at the end of the day, your final product is this, an activated tRNA. When you say activated, it already has the amino acid. So you call it tRNA activating enzyme. After it binds amino acid, it's now called an activated tRNA. Okay? Now, stages of translation. Does this sound familiar? Initiation, elongation, and termination. Similar to transcription. Okay. For transcription initiation, you look for the promoter region. For translation, this is what initiation looks like. The small ribosomal subunit, the small first binds. Okay, just the small. The small first binds. And then, see how the small started from the 5 prime end. It looked for the 5 prime cap. So when it saw the 5 prime cap, it started reading it from the 5 prime end. And it's going to slide down the mRNA until it locates the start codon. Our start codon happens to be, I told you I can't forget this because my birthday is also August. So it starts when it sees the AUG sequence. So please, in your brain, don't think 5 prime is always followed by AUG. There can be other sequences there. And then it finds AUG. When it finds AUG, the tRNA and the large ribosomal subunit, so imagine this is not yet here. When it finds AUG, the complex joins in. So the tRNA will now bind together with the large ribosomal subunit. Now, to confuse you a little bit, or maybe not to confuse you, never mind, I don't want to confuse you. So small ribosomal subunit first, it looks for AUG. Once it finds AUG, it signals the first tRNA and the large ribosomal subunit to join the complex. The AUG codon has UAC as its anticodon. And UAC always carries methionine. So the start codon always starts with methionine amino acid. So all of your proteins always has methionine at the front. Okay? And very important to remember, the first tRNA will go to the P site. The P site. Okay? The next tRNA will go to which place? A. So the next tRNA will go to the A site. And then after that, it moves along the next codon. This goes to the E. The A goes to the P goes to the E. Just like in the animation. So it moves through these three sections, from A to P to E. All of your incoming tRNAs goes to the A, then the P, then the E. Except the first tRNA. Where does the first tRNA go?
P.
The P right away. The succeeding tRNAs will all go into the A, then the P, then exit at the E site.
Miss, you said the beginning is IUG. What's the end where it stops moving?
Later. Thank you. Yeah. We also have specific codons to remember for that. All right. So ignore everything here. That's exactly what I just said. Remember, there's three sites in the large ribosomes, A, P, E. The first tRNA goes to the P. That's all you need to know here. And then also, you see in the animation, once they're in the P and in the A, right here, P and in the A, those amino acids, because they're side by side, they can form peptide bonds. Here. They can form peptide bonds. The tRNA leaving the E site does not have the amino acid anymore. It left it bound to the amino acid of the tRNA found on the A site. Yes.
So the polypeptide forms on P.
Yes, the elongation is here, between A and P.
So,
Incoming bind. When it moves here, no more.
so it loses the amino acid.
It loses the amino acid in the E. Good question. It loses the amino acid in the E. So at the P, it leaves it there. It leaves it at the P site. Then it becomes longer and longer. Until, so many. What else do I have to say? Not too many words, miss. Too many words. Until it reaches, this is your question, Swane, stop codons. So, if your ribosomal complex reaches either, so it doesn't need to be these three, either UAG, UAA, or UGA, then the complex will dissociate. It will stop translating.
Does this also apply to DB and TPs?
No, because the NTPs is replication. This one is a complex that the whole complex stops doing its process. For DDNTPs, it prevents the polymerase to add more. But same concept, it ended. Like you can't further translate the whole stretch of your mRNA. Easy, right? Filipinos, again, I have like a very funny way to remember this. Uwag means wag. Uwa means baby is crying. Uga in Filipino is to shake. So uwag, baby, uwa. If you shake it, uga. Wag, uwa, uga. Find a way to remember that.
Okay.
Or find some other mnemonic in your brain. Because like UA, I don't remember that.
What starts with U
You?
and it has ING.
Yeah, but then the order.
It just-
Maybe
you
like
know,
this one.
there's AGC.
Ooh, UAG and then switch UGA.
Yeah.
And then double it, UAA. Yeah, with your 6 IB subjects, you won't remember. Okay. Now, where does this happen? In the cytoplasm. Where in the cytoplasm? It could either be in the free ribosomes or the ribosomes in the endoplasmic reticulum. So where does it happen? Animation first, then I explain. A newer edition, what? Okay, my animation is not working. I'll just explain. Here's how it's gonna happen. So as you are, so this is your mRNA, small ribosome, large ribosome. Are you comfortable with that? Don't tell me, Miss, why is that in the bottom? Why is that at the top? Does it matter? It can be turning. Okay, so it's reading your mRNA, right? So this is now the growing peptide chain, polypeptide chain. Now there's what you call a signal sequence. Miss, what's a signal sequence? It's just a sequence of a polypeptide that triggers your SERPs or your SRP, signal recognition particles. These are proteins. Look at what's happening here. I hope in your brain this is moving. This, when it finds the signal sequence, it's going to grab this complex and pull it here into the endoplasmic reticulum. So it's going to pull it to the endoplasmic reticulum because it knows that this protein that you're translating is destined to be going to your Golgi or to be put in a vesicle to be released by the cell. So look at what's happening here. So it pulled the complex to the endoplasmic reticulum, and see how it continues translating. Now your product, your protein is now inside the RER. Oh, that's cool, miss. That's why from the RER, protein is sent out as a vesicle into where? The Golgi. So this, imagine put it in a vesicle, and then endocytosis is going to go into your Golgi. And then in the Golgi, it will be folded, it will be packaged into the final protein that is needed by the cell.
So the SRP pulls it in?
Yeah, into the endoplasmic reticulum. So this is another group of proteins that can pull your complex into the endoplasmic reticulum. All right? Sketch this. There's no other way to remember this into words. Like this, pull. If there's no signal sequence, then it's just made in the free ribosomes. It's just made in the cytoplasm. That protein will be used inside the cell. DNA is so unique. The sequence also tells the cell whether it's going to be protein destined for the outside of the cell or used inside the cell. Very cool. Yes.
At the end, it separates, right?
At the end, yes.
So that mRNA can be used again?
Yes, very good question. mRNA are RNA nucleotides, right? If this is done, enzymes will act on it to break it into free nucleotides again. That can go back into the nucleus and be reused for transcription. We are just reusing
So it just
nucleotides.
reuses the whole
Yes, yes.
So it doesn't make any new ones?
Remember you told me earlier, there are junk DNAs
Oh, yeah.
that can also make new RNAs or tRNAs or mRNAs. So you can.
Oh, okay.
But you can also recycle. Really nice questions. Every year I have different questions from different people. Okay. So here's how it's going to look like now. Vesicle buds off the membrane, and this will go into your Golgi. Fancy. Tell yourself, remember, SERPs, SERPs, SERPs. Here's another illustration. So if there's no signal sequence, the protein will be directly used inside the cell. Maybe it will go in the nucleus, mitochondria, or the chloroplast, or the peroxisome. But if it's pulled into the endoplasmic reticulum, you know that this protein is destined for the plasma membrane inside the secretory vesicle, or it can be a protein used by your lysosome. I don't want you to be confused there. Lysosome is a one membrane organelle, vesicle, lysosome. Vesicle has protein. It fuses with the lysosome, so the lysosome increases in size. Why does it need to give protein to the lysosome? What is the lysosome for? Digestive enzymes. Enzyme is a protein. So it can also go into your lysosomes. It's protein, really amazing. They're everything. They're everything. Okay. So just a note here. For prokaryotes, as soon as you have an mRNA, you can translate right away just because they don't have a nucleus. There's no barrier. For eukaryotes, it needs to travel from the nucleus to the cytoplasm. That's just an idea you need to know. Okay? All right. Now, your proteins. Your proteins can come in different structures. If I talk about the primary structure, let's say, okay, protein A. What's the primary structure of protein A? I'm asking what is the sequence of the amino acids. So whenever I ask the primary structure, not whenever I ask, whenever you're asked, or when there's a question about the primary structure, you're basically asking what's the sequence of the amino acids. The sequence. So primary structure is asking about the primary sequence. What are the order of the amino acid? You already know. If you change the order of your amino acid, it becomes a totally different protein. The order also dictates the kind of protein. So primary structure just tells you the sequence. Secondary structure. This is really cute. Secondary structure is you have the sequence and the amino part of one amino acid can react with the carboxyl part of another amino acid to form hydrogen bond. See, this is nice. You've learned all of this. Hydrogen bond. How is this a hydrogen bond? The H is bound to an N. Remember NOF? So it means it can bind to another area here in your amino acid. In this case, the carboxyl N. Remember carboxyl is COOH. It also has an H bound to a NOF, an oxygen. So you can form hydrogen bonds here between the amino group and the carboxyl group. I don't want to fast forward that, but you should remember the structure of your amino acid. I'll just draw here for you so you'll remember. Remember that? So this H can form hydrogen bond with this of another amino acid. Remember, there's an OH and then there's an O. So this can react with this, release as water.
So
And you have, yes, and you have a peptide bond. Oh, no. You have a, not peptide bond. Peptide bond is between amino acids. This one is between side chains, like the amino group and the carboxyl group. It's not a peptide bond, we call it a hydrogen bond. And hydrogen bond is not as strong as a peptide bond. Although it's the strongest intermolecular force, you can still break this hydrogen bond. Sketch in your notebook so you'll remember. But if you understand just by the picture, that's good. Amino of one amino acid can form hydrogen bonds of a neighboring carboxyl of another amino acid. Therefore, if all of those hydrogen bonds can form, your primary structure can actually start to form like a twisted structure. And if it's like a twisted helical spiral structure, we call it an alpha helix. Okay, secondary structure can either be an alpha helix or a beta pleated sheet. Now let's look at the beta pleated sheet. This one is helical, this one is folded. See how this is also between amino and carboxyl on the other side. So it can form folded pleats. Do you know what pleats are? Your skirt has pleats. So beta pleated sheet. The protein secondary structure can look like it's folded. So it's either it's twisted or it's folded. And if I ask you why, miss, so easy. Because hydrogen bond miss. Between what? Amino and carboxyl formed. Those intermolecular forces formed. So your protein now is helical in structure or folded in structure, or it can even be a combination of both. For example, here, I have here, oh, this one is not combination. This is myoglobin. I think I use this as an example because it's nice to know that you have a molecule called myoglobin. Myoglobin is similar to hemoglobin. In fact, they sound the same, myoglobin, hemoglobin. What does hemoglobin do? It's found inside your red blood cells, carries oxygen. Now, we have a molecule similar looking to your hemoglobin. It's found in your muscle cells, inside your muscle cells. These myoglobin, they don't spread around your muscles like how blood can travel around the body. It's just in your muscle cell itself. And they can hold oxygen onto their molecule. Therefore, if there is not enough blood supply to your muscles uses your myoglobin as a source of oxygen. Okay? So just nice to know. And as you can see, look at the structure. Secondary structure, it's alpha or beta?
Alpha.
Alpha helix. So clear there. Alpha helix.
Yes.
Mm-hmm.
Does the myoglobin have something to do with the lactic acid you're not?
No. Lactic acid just means, well, kind of. It means you've exhausted your myoglobin's oxygen as well. All right. Now it can be a combination. This is a very timely example. This is a structure of an immunoglobulin. Oh, so fancy. In English, antibody. Now, if you look at a structure of an antibody, it's made up of combinations. There are helixes and there are pleats. There are helixes and there are pleats. So combination of either an alpha helix or a beta pleated sheet. There are several secondary structures for your antibodies. Okay? All right. Now let's go to the next structure. So your primary structure, just the sequence. Secondary, either spiral or folded. Tertiary now, tertiary. Before we start with the tertiary, look at this table of amino acids. Remember you have 20 amino acids. We can group them according to their chemical properties. So we have nonpolar amino acids. Oh, you just learned what nonpolar means. Polar. Oh, you learned charge. It says uncharged. It's polar and there's no charge. And there's positively charged. Maybe this is 1 polar. In fact, ionic, maybe closer to ionic, like a dipole-dipole. Negatively charged, and then you have the nonpolar aromatic group, those with rings. Okay, Miss, why do you show this? I wanted to show you this for you to understand that, remember the R group here, the R group can look different. There's 20 different kinds of R groups here. See the bolded, the shaded blue? These are the R groups. Now, if you have all of those R groups side by side, they can form bonds. Here's an example. If you have the hydrophobic, your nonpolars, hydrophobic, yeah, right? Nonpolars, and then there's another nonpolar side chain there. It can form a hydrophobic bond here. It can pull. Or it can fold, not pull. It can fold your protein. And this is nothing new, hydrogen bond. We've seen hydrogen bond in secondary structure, right? Those side chains can form hydrogen bonds. So, fold again. This is what you need to remember. The disulfide bridge. Sulfur bonds can form. Sulfur of one with sulfur of a neighboring amino acid can fold your protein. This is a very unique folding for a tertiary structure. Sulfur bridges or disulfide bridge. Sulfur bridges or disulfide bridges. And then another example here. Remember you have the charged. So charged and a positively charged or negatively charged can be attracted to form ionic bonds, very strong, even more folded. Okay, so in your brain, those side chains can react to fold, to fold, to fold, to fold. your protein. Those, yeah, like clumping, right? Compacting. There's so many folds there. It's because the side chains are interacting with each other. They can form hydrophobic interactions. They can form hydrogen bonds. They can form, very important, so for bridges, they can form ionic bonds. All of that results into clumping, folding of your protein structure. So, does this look familiar? Helix, right? So helix with beta pleated with further folding, you're talking about globular proteins now. Globular proteins are like globules, like enzymes are globular proteins. So many of your all of your enzymes are in its tertiary structure. They have a specific folding, they have a specific shape for their active site. If I break this bond, which is called protein, nature. If I denature those bonds, you change the folding, therefore you change the shape. Therefore enzymes will not work.
Miss, what's
Yes.
the in the active site of the enzyme? Like what type of, like for DNA you have the A, C, C, that stuff, right?
Our matching.
Yeah, how
For
does it work
enzymes,
for
it's
enzymes?
just the shape. And it's a theory. We call it the lock and key model. It's just a theory of how enzymes work. Maybe it doesn't even work that way. We don't know. But for now, that's our model. We're saying it's the shape. Because when we change the shape, the enzymes seem not to work. But we don't have a matching like this, quote unquote. It's just really the shape. Yes.
So enzymes are mostly in the tertiary.
Enzymes are tertiary proteins.
So
They have to have a specific shape. If you say enzymes can be in secondary, that's hard to understand because how can you give shape for its active site? So all enzymes are tertiary proteins.
So the more heat, the more bonds it can form.
No, the more heat, the more bonds it can break. It's folded already. This folding, where do you think happens? Where do you think this happens? You were just talking about the endoplasmic reticulum. In the Golgi. In bio, we say the protein is packaged or modified. In chemistry, we understand it being formation of these bonds. That's why it's packaged. It's being folded in the Golgi. This can also happen in the cytoplasm, of course, if the protein will be used outside just inside the cell, yes?
So it's like, for example, the protein goes through the Golgi and it forms, and then it can go out the cell and act like an enzyme?
Yes. You can also, your body's cells also releases enzyme, like digestive enzymes, right? It's released by the cells of your small intestine, the cells of your pancreas. And then we're almost done. Quaternary structure. This is when the protein is made up of several subunits. What is your histone again? 8 subunits. This is a classic example of a quaternary protein structure. It's made up of several subunits, or like several smaller polypeptides. As you can see, this is a different polypeptide than this, than this, than this. So you have 4 translations. Think with me, please. Translation of this polypeptide first. Translation of another polypeptide. So these are products of 4 translations. Okay? Our example here is hemoglobin. Hemoglobin is made up of 2 alpha and 2 beta polypeptides. So the beta looks similar, alpha looks similar, but again, these are products of separate translation processes. And they combine together to form your quaternary structure. You need to know, this is the last slide. Some proteins will have a prosthetic group. In medicine, are you familiar with prosthesis or prosthetics? Like you cut your feet and then they put like a metal leg. That's called prosthetics. The same idea here. When I say prosthetic group, you're adding a non-protein molecule to your protein molecule. Okay? For example, here. This one, what is this?
Iron.
Iron. Iron is an element. Iron is a metal. Iron is not an amino acid. It's not part of a protein. So with another with another non-protein, we call it we call that non-protein as prosthetic group. Wait, I think there's a name for that. My brain. Quaternary with a prosthetic group. Can you read through this quickly? Anything there that says quaternary with a prosthetic group?
Where does the iron come from?
Hemoglobin. For example, I get apple. I eat apple. There's iron in
It's
apple.
from food.
Yes. Those micronutrients are from your food. Your body
So your
can't
body
make.
can't.
Yes, correct. Your body can't make it. That's a good question. So like zinc.
That's why you
Iron.
have to take like
That's why you take vitamins. Yes. Because you can't code for those elements. There's no word.
Just says heme groups.
Yeah. In groups. Maybe I'm just overthinking. I don't know. I think there should be a keyword that I'm not saying yet. It's okay. It's gonna come back when I think about it. All right, just nice to know here. You have a picture. You have a diagram of collagen. Collagen are found under the lining of your skin, so it keeps your skin like looking like young, keeps it in, like stretched. If the older you are, those collagen molecules degrade. So your skin becomes loose and then you form wrinkles. That's why people put like collagen whatever in their face. That's it. Any question?
Are you gonna post the
Video. I will post the video.
slide?
Maybe. Yes, maybe. I don't know. I hope I don't forget. Your homework, children. Wait. Huh? Did I not record it?
oh wow.
Oh, no. Stop screen.